Welcome to Life and Biology. I'm Dr. Joel Graff, and the next couple of videos are going to focus on the trp Operon and the lac Operon. Now, these are classic examples of genes that are regulated within a bacteria based on some certain circumstance. So you would hope that if a cell is going to control what genes it's going to be regulating, that there'd be some logic to it. So I remember learning these operons back when I was in college, and what I remember was that it seemed confusing and it seemed like I could easily switch around a couple of components in the systems and then end up with the wrong answer on my test. So what I'd like to do with these videos is to try to talk through the logic behind whether the genes are being regulated one direction or not, being expressed or being silenced. And by talking through the logic, hopefully that can just make it easier to remember all of the steps that could control it. So the first example in this video is going to focus on the tryptophan operon. And the thing about the tryptophan is that it's an amino acid. And you might have read on a cereal box or some other food packaging lists of amino acids, or essential amino acids, and non-essential amino acids. That's kind of hard to say for some reason. But anyway, if you have essential amino acids, it means that the genome of the organism that is consuming the food doesn't have enzymes for making that particular amino acid. If you don't have the enzymes for making an amino acid, then you have to eat the amino acid, or you don't have that particular amino acid. You can't build all the proteins you might need to build, and you are sick or dying. So then if you have essential amino acids, then there must be something called non-essential amino acids. And if the definition for essential was that you don't have enzymes, the essential for non-essential, or the definition for non-essential amino acids must be that you do have some enzymes. And so with those enzymes, you can take precursor metabolites. These are just any compound in a metabolic pathway is called a metabolite, and they can be converted into whatever amino acid it is that you're talking about. Okay, so with tryptophan, we're dealing with a non-essential amino acid, and it means that, and we're going to be talking about bacteria, but if the bacteria has access or has plenty of tryptophan around, there'd be no need to make any tryptophan. And if there's no tryptophan around, then the bacteria might want to make some enzymes so it can convert other molecules into tryptophan. Okay, so that's kind of the logic here. Now, we've got a metabolic pathway, a real generic one, where metabolite A goes to metabolite B to C to D to E. And for each of these changes in the compound, whether we're adding something on or taking something off or rearranging the atoms that are in that molecule, we need an enzyme, or at least to get the process to go fast, you need an enzyme. And so I have some generic enzyme names listed here. The first enzyme is E for enzyme AB, meaning it would take metabolite A and convert it to B. And so then I have 3 more enzymes listed here that take one metabolite and convert it into the next metabolite. So let's say E in this generic example represents tryptophan. If the cell has plenty of tryptophan, you don't need to make any of those enzymes, which would be a resource-intensive thing for that bacteria to do. So if you have plenty of E around, don't bother making any of those enzymes. If you don't have enough E around, then you might want to make these enzymes. And if you have metabolite D, you only need the enzyme EDE that will convert D to E, because that will get you to tryptophan. If you have all of these enzymes being expressed, it means that whether you have molecule A, B, C, or D in the cell, if all of these enzymes are present, they could work in tandem to make the tryptophan for you. Okay, and so that's the situation we have with bacteria, is that they have all of these enzymes regulated in a coordinated fashion, so that if there's no tryptophan, no end molecule, you would make all of the enzymes that could lead up to that final molecule. You have those all regulated together. So when a bacteria coordinates a series of genes all at the same time, that's called an operon, along with the sequence in front of the transcription start site that controls whether the gene's on or off. So here's an operon. We've got a promoter, an operator, and then there's a transcription start site here, and then you've got 4 different genes, and I just labeled them A, B, C, D. It represents trp A, trp B, trp C, trp D, trp E. So those are the 5 enzymes that are all coordinated by this one little stretch of DNA. So a lot's going to be happening at this stretch of DNA to control whether you turn those enzymes on. All right, so make only what you need. Well, I've set up a situation here where blue dots up here represent tryptophan. So in this case, the cell has plenty of tryptophan, and you wouldn't want to transcribe these enzymes. It would be a waste, and maybe those precursor molecules could be used for something else. So you want to block the transcription. So I drew a blue X through the transcription start site arrow. And the reason that the transcription is stopped is not because we don't have a polymerase being recruited. The polymerase will just automatically be recruited to the promoter. There might be something more than that to some of these stories, and we'll give an example with the lac operon. But let's just say the polymerase just automatically gets recruited to the promoter. Then you've got the next sequence between the promoter and the transcription start site is the operator sequence. And so the operator has a specific DNA sequence that can be bound by a protein that can bind a specific sequence of DNA. So this repressor, whatever it is the type of protein that can recognize and bind to the sequence that whatever that sequence is right there in the part that we're calling the operator. Okay, so that repressor can bind to the operator. You'll notice I drew it a little bit Pac-Man shaped, and I put in a little dot there, and as we can see, that dot represents tryptophan. This is, we talked about enzymes, whether they can be active or inactive depending on whether they had the cofactor. Well, these repressors, they kind of follow the same theme. Whether they're bound to a molecule or not can change whether they can bind to DNA or not. So in this case, the repressor can bind to the tryptophan. And when you have tryptophan sticking to the repressor can stick to the operator. And that creates a nice roadblock so that you cannot have the polymerase go through that sequence and get to the transcription start site, okay? So a repressor bound with tryptophan is a nice roadblock for the polymerase, and you don't turn on the expression of all of those enzymes that were controlled at that operon. Okay, so here I don't have any tryptophan blue dots being shown, but I do have a lot of blue circles. So what's going on? Well, because there's no blue dots sitting in that site on the repressor can't stick to the DNA. In that case, the polymerase gets recruited to the promoter, and then it works its way down the gene to the transcription start site. It makes the RNA, and then those RNAs are made into proteins. Okay, and so we've got these genes turned on. We're turning on these genes, trp A through trp E, because these enzymes can convert precursor molecules to tryptophan. So once you get these enzymes being made, we're going to be able to, I didn't draw it on this picture, but let's assume that there were some precursor molecules. They can be acted on in a particular order by these trp A through E genes, and it'll make tryptophan. And if we build up enough tryptophan, we can go back to this state where we have tryptophan, and that's when you want to turn these sets of genes off. So to summarize, make only what you need. If we have tryptophan, don't make all those enzymes so that you can make more tryptophan. If you don't have tryptophan, then get that repressor out of the way, and you will get the polymerase to scroll through and transcribe all of those genes, and the mRNA will be made into the proteins for all of these enzymes in the tryptophan pathway. So that's an example of where a cell would need to make more of a particular molecule and wants to turn on the expression of the genes that could make that molecule. All right, we'll switch over to lac operon in the next video, but hope that was helpful. Just think through the logic of your drawings there a couple times, and that should stick. The lac operon's a little bit more tricky, and there's more options of than just whether tryptophan's there or not. So anyway, if you like this video, you can subscribe or not. Talk to you later.